Hi, my name is Amy Lee, and I'm a senior at the University of Wisconsin-Madison. And I had the amazing opportunity to participate in the NIH SenNet CUSP internship program at the Adams Lab at Sanford Burnham Prebys in San Diego. So aging happens to everyone and is a process with a lot of different factors involved. There are a few key mechanisms that affect how we age, which are called the hallmarks of aging. One of these is cellular senescence. These senescent cells will accumulate in tissues as people age and will contribute to normal aging process and age-related disorders. And so what is cellular senescence then? A senescent cell is characterized by stable cell cycle arrest, but it's still metabolically active but resistant to cell death. And so it can be caused by telomere erosion, DNA damage, epigenetic changes, oncogene activation, and organelle stress. When a cell is arrested, it'll release different proteins known as CASP or senescence-associated secretory phenotype. And SASP is a characteristic of senescence that will lead to different effects in the physiology, which can be good as it can cause tumor suppression and wound healing, but can also lead to pathology, which is bad, and lead to chronic inflammation and tissue dysfunction. So P16 and P21 are important because they are canonical markers of senescent cells. They are both tumor suppressor proteins and cyclin-dependent kinase inhibitors, and they both slow cell division by slowing the progression of the cell cycle from the G1 phase to the S phase. This is an overview of the San Diego Tissue Mapping Center. The focus is to spatially map at the single cell level the aging of the liver, brain, colon, mammary gland, and bone marrow during aging. So these 5 tissues are processed and they're sent to bioanalysis core, where they use multiple approaches to spatially map these senescent tissues. The approaches are shown here. And then it's sent to data analysis core before it's given to us, where we get a list of genes that are involved in senescence. And those are the genes we knock out for functional analysis, but also can be used as validation for other techniques. So this is where I'm involved, and we'll use CRISPR to knock out these functional genes. Like I mentioned, we use CRISPR. So CRISPR will be specific to our genes of interest, and we use guide RNA. It's designed to be complementary to a specific DNA sequence in the genome. So Cas9 is recruited, and it'll bind to the target DNA, leading to a double-stranded break. Incorrect genome repair will lead to the gene being knocked out. So we use AAVs to deliver CRISPR into mice, and AAV stands for adeno-associated virus. It's a linear single-stranded DNA genome of approximately 4.7 kilobases. And so AAVs will bind to a specific cell receptor on the surface of the target cell. It'll go into the cell via endocytosis, and the virus will escape the endosome into the cytoplasm. The AAV particle will travel to the nucleus of the cell, where it'll uncoat and release a single-stranded DNA genome before it's converted to a double-stranded DNA. So the delivered DNA can then be transcribed and translated by the host cell's machinery, leading to the production of the desired protein. This is the overall CRISPR pipeline that we use. So first, we want to design the guides and clone them, and then test these guides in vitro. So we want to do transfection, then gDNA extraction and PCR, which gets sent for sequencing. And we analyze it using ICE, and we'll see if our knockout worked. And if it did, then we want to continue to test these guides in vivo and grow these plasmids for AAV production before we inject them into the mice. These are some results that we have from the in vitro process of the experiment. This is after our PCR gets sent for sequencing, and this is the analysis. So we have our control locus, which is ROSA 26 and P16, and these are discordance plots. The y-axis is discordance, or a level of disagreement between the non-edited wild type or control and the edited sample in the region around the cut site. And our x-axis is the Sanger coordinates, which is the nucleotide positions in the DNA sequence being analyzed. We can look at the control first, and we see that the green line is the edited and the orange is the control, and this entire area, they are overlapping. So we know that our control is working and the sequence hasn't been changed and P16 hasn't been knocked out. If we look at the P16 graph, we see that the edited line in green is above the control line, which is orange on the bottom. And so because these two lines are not overlapping, there is a level of discordance between the two. And we know that the sequence has changed so that P16 is knocked out. This is the same control we use, except we're looking at P21 instead. If we look at the P21 graph, we see that on the bottom line in orange is the control and the top is the green line edited. And we know that once again, there's a level of discordance between the two. So we know that P21 was knocked out and the sequence has changed. And so overall, in the CRISPR pipeline, again, I've been working on the in vitro part, which is the in vitro part, and as well as a little bit of the in vivo part with growing the plasmids. But P16 and P21 are just some examples of the guides we've done. We've done many more, but other members in the lab have continued to do the in vivo process. And so one of the ones they've looked at is P53, which is a tumor suppressor, and it's upstream of P21, so one of our targets of interest. We are conducting functional analysis on the mice. So this is a Western blot we have here. And on the left is the control. So P53 signals are here. And on the control, we see that there are signals for P53. So we know that P53 was not knocked out. On the right, we have our knockout of P53. There are no signals of P53 on the right, so we know that our knockout worked. We were looking at DNA in vitro before, but now we're looking at the protein level in vivo. So this proves that our model knockout gene works at the DNA and protein level. So in aging, we see an upregulation of P53. So if we target P53 and knock it out, we want to see how it will affect the aging process and senescence. And so some future directions we could take would be to get more data from the SD Tissue Mapping Center that'll tell us which genes to target next. We could also generate guides and capsids that target other tissues of interest, because currently we're only looking at the liver, but we could also target other tissues like the mammary gland or the colon, like I mentioned previously. We can also knock out genes for functional assays in vivo and validate other techniques using knockouts. In closing, I want to thank the Adams Lab, including the PI, Dr. Peter Adams, Carolina, Marcus, and the rest of the Adams Lab for creating such a welcoming environment. I also want to thank Kay, Elizabeth, and Natasha, as well as NIH SenNet, for giving me the opportunity to be part of this program. It was a really great learning experience, and I know I'll use the skills I gained in the future. Thank you.